Hi, I'm Anjeanette Roberts, and I'm a reason to believe research scholar. And my background's in molecular biology and cell biology, but more specifically in virology, virus research. And most people around here call me AJ. And I'm just coming on today hoping to give you some encouragement and some information that can put your minds at ease and just give you a better understanding of what's going on, address some of your specific questions related to COVID-19 or the SARS coronavirus 2. In my research experience, I actually spent 3 years at the National Institutes of Health, where I was working on SARS coronavirus, which was causing a much smaller outbreak in 27 different countries in 2003. And so I spent quite a bit of time researching animal models and potential treatments and vaccines for that coronavirus, which is actually so similar to this coronavirus associated with the COVID pandemic that they've named this coronavirus SARS coronavirus 2. 80% similarity. And so I know quite a bit about the original SARS, and I know quite a bit about this one as well. And so hopefully the things that I know can answer some of your questions and can give you some ways to think about the things that can be pretty overwhelming and disruptive right now as we turn on the news and we look around and we're uncertain about so many things. But there are reasons to believe we also, as you probably know if you're tuning in today, that we look for ways that science and faith can be deeply integrated. And we believe that the things that we know about nature and creation will only reinforce a biblical view of the God who loves us and seeks us out in the person of Jesus Christ. So thanks so much for joining me today. When I talk about this topic, I really like to talk about it in sort of four categories. One is to love boldly. The other is to serve wisely. The third is to think clearly. And the fourth is to pray always. And so I'm going to address questions hopefully in all four of those categories in the next hour. But I really want to start with the love boldly, just to encourage you that we're all feeling very vulnerable and very overwhelmed. And small actions of encouragement and reaching out and connecting with people, even virtually like this, can really go a long way to help people feel safe and loved and less fearful and less insecure. And so love boldly. But when you love, serve wisely. So practice all of the personal hygiene that you've been hearing about. Wash your hands with soap and water to a lather for 30 seconds. Use warm water. Do not just personal hygiene, but environmental hygiene. And then maybe equally important to loving boldly is to think clearly. And so one of the ways that we can also contribute to a sense of not feeling overwhelmed or fearful is to try not to share misinformation. And so that's what I mean when I say think clearly. At least when you're seeing something on social media or you're having conversations or something comes up on the news, maybe pause and ask yourself these two questions. What's the source and what's the evidence for what this person is presenting as factual? And I think that those are two really good questions to ask all the time. And so hopefully we'll be able to help you think clearly, not just in the next hour, but we have all kinds of resources available at reasons.org. I blog there as well as several of my colleagues. We have a COVID-19 dedicated page or coronavirus dedicated page there. So you can check out those resources as well. And then, of course, you know, we are a Christian organization. And I think that we should always remember Paul's words of encouragement to pray always, because God's at work when we pray. And so love boldly, serve wisely, think clearly, and pray always. Now, I'm going to turn and take some of your questions. I don't want to spend a whole lot of time just talking about me and my expertise. I want to talk about what's interesting to you. And for that purpose, I actually like connecting with people and not just talking into a digital world or virtual reality. And so I've invited my good friend and coworker, Anastasia Young, who is the publicist here, and she is always hooking us up for events like this and radio and interview. And so sort of turning the tide on her a little bit this morning and asking her to jump in and do this live Q&A with me. So hi, Sia, Anastasia, welcome.
Hey, AJ. I am so jazzed that you invited me to do this. especially because before all of this happened, and before the lockdown, let's say, when we went to our respective homes, um, it was such a treasure to have you in the office, and be able to have those doorway conversations with you, and draw on your understanding, but also your ability to put the cookies on the bottom shelf of like what's happening. And this is just fantastic that we get to actually share you with everybody. And got questions already lined up. They're already coming in. So if you don't mind, we'll just jump right in.
Let's do it.
All right. All right. So we have Rob Welton. He asks, when will we know that plasma from healed corona victims will, one, keep them immune from the same virus, and two, be able to help those who currently get sick to help them get better?
Yes. So thanks, Rob. We actually already know the answer to the second question. That it's called convalescent plasma. Convalescent means somebody who's passed through the disease and they're no longer carrying the virus, and they actually have in their blood antibodies that neutralize, that are specific to the virus. And neutralize is the scientific laboratory term for when you add the antibody to the virus. It's a sterilizing immunity. It prevents the virus from actually being able to infect the cell by blocking the viral receptor from the cell receptor. And we know that neutralizing antibodies to this virus are protective. Those plasma transfers of convalescent serum have already taken place. They were taking place in China early in the outbreak, and it is proven to be a very effective treatment for people who need it. But as you might imagine, it's a very limited resource. And until we can test people, serologically test people for those antibodies, and determine that they've recovered from the infection, and also that they're negative for virus, that the virus in fact has been cleared from their system, then we can't. It's a limited resource because there's only so many people that have met that criterion. But until we have widespread serological testing, we can't tell if there are more people out there who could be doing that because they recovered from an asymptomatic infection. And possibly up to 50% of these infections may be asymptomatic. And so the first part of your question, Sia, could you repeat that for me?
No, I can't. I can't because they erased it for me. Oh, here it is. Wait. The first part of the question. Here we go. So how will that keep them, the person that originally was sick, how will it keep them from getting the same virus again?
I remember now. Yes. So we also know from data from the first SARS outbreak in 2003 and from the data to date that those neutralizing serum antibodies persist. But they probably are not a durable immunity. And so the question becomes, how durable? How long will somebody be protected if they have those antibodies? And it's really very early, relatively speaking, in this outbreak to be able to determine that, because typically those antibodies will persist for several months. And so we're only, still only in month 4, maybe the beginning of month 5 for this pandemic. And so it's too early to say how long will they last, because everybody that we can measure today probably still has a durable immunity. We could compare it to some other coronaviruses, the ones that cause upper respiratory infection. And from data from those types of infection, we know that people can become infected perhaps as frequently as every other year. And so, but there's been intermittent infections with other coronaviruses. So we don't know if that sort of resets the immune system or the immune profile. So I would say, yes, people are safe. They're probably safe from reinfection for at least a year and a half, possibly 2 years, probably, and possibly even much longer. But there is some data that would argue sort of the opposite of that. There have been cases of MERS, which is another coronavirus associated with the same type of really severe lower respiratory infections and a much higher fatality rate. And people who have recovered from MERS, their durable immunity has lasted only about a year, and maybe just slightly under that. So we really don't know until we see the actual measurements for this virus.
I know that I've seen that question myself, on Facebook, where I get all of my information from, because that is the most reliable source, I'm sure.
Well, it's always a mix, isn't it?
It's always a mix.
There's
But
some
no,
not so good stuff, and it's having to weed through those things. But I post a lot of things to my Facebook page, and I fact check everything that I post. And so, and if I get my facts wrong, people call me on it pretty quickly, and I'm pretty quick to make a correction. But we have to remember that this is an evolving situation, and it has been from day one. And the best that we can do is give you our best information today, and that's true of anybody that you hear in the medical and scientific community. It's why I think some people are getting frustrated with the different models. They're like, hey, wait a minute, how can you keep changing your mind? Oh, we're not changing our mind, we're getting more data, and the data is revising the way that we understand the pandemic and the way that we understand the best way to control it.
that is very helpful information to just a good lens to look at all of this through. That this is the best information we have right now. This is how we're moving forward, and yeah, we'll just continue learning as we go. The next question comes from, and this name is just it's profound because they call themselves petrified, and God bless them. I mean, I know a lot of people are in this position. So Petrified asks, if everyone wore a mask in public and washed their hands frequently until there was a vaccine, could we at least partially get back to a normal life? Example, get a haircut, see a movie, just some sort of normalcy.
Yeah, I actually think that that would go a long way to working. If everyone washed their hands frequently and didn't touch their faces and everyone wore a mask, we could probably get back to a new level of normalcy pretty quickly, if you consider that normal. Washing your hands, hopefully that is normal. Wearing the mask, some people are getting pretty creative. But the masks, again, they're not so much to protect you from being infected. What protects you as much as you can from being infected is being around other people who have masks on, because the mask is helping to contain any particles or droplets that might be coming out as they breathe or speak. And if you think, well, they're not coughing, certainly I can't catch anything from somebody who's not coughing. Well, that's not true. Just take a mirror or take your cell phone and hold it up to your mouth while you talk. And as soon as you pull it away, you see that there's moisture on the phone. Well, those are small respiratory particles that are coming out of your mouth. And so the mask is meant to do just that. It's to keep those respiratory particles from coming out and getting closer to you if you were in the same room with me, right? So it's not so much protecting me because you can get it through breathing in droplets, but you can also get it through contacting contaminated surfaces where droplets have basically fallen and dried. And then you can get it not just through your mouth, but you can get it through your nose, through your eyes. SARS is a virus that is not like flu in that it's just spread through respiratory droplets and where those land, but SARS can actually be shed in many different bodily fluids. And so you could isolate SARS from tears, from saliva, it's been detected in sweat, in blood, in urine, and also in feces. And so SARS is a lot different than flu, even though some of the initial cases and some of the worst outcomes can look similar.
Wow, that's the first I've heard about tears. I mean, it makes sense. But so with what you're saying about the breathing and washing the hands, you feel that if we took these precautions, these reasonable precautions of wearing the mask and washed our hands and maybe social distance, didn't touch our face, social distancing, that perhaps we could get to some sense of normalcy, it sounds like.
I think so too. And I think the longer we all stay in stay-at-home mode, the more the question that Petrified asked is going to become relevant. When can I get my next haircut, right? Because getting your haircut is you're pretty close to other people, and so they're washing their hands as they're washing your hair, and as long as neither one of you is touching your face and your hands are clean and you have that mask on so that you're not contaminating things with the breath that you're releasing in case you're asymptomatic or your hairdresser's asymptomatic or your barber's asymptomatic, then activities like that should be safe. But I don't know about you, but even here in California, where we've done a great job of really flattening the curve and keeping things slow so that our health care system thus far has been able to manage things well, and we're not near our peak yet, and so that's good. We're doing what we need to do. Governor Newsom's done a great job telling us all to stay at home. But even when he was telling us to stay at home, I've got to tell you, driving around in Los Angeles County, I saw plenty of people who weren't physically distancing. I think we're doing better now. I'm seeing lots more people with masks on, but human behavior modification is one of the hardest things to accomplish. So how do we ensure that people are wearing masks? How do we ensure that people are washing their hands? If we could have a society of trust, that would work, right?
As Christians, we know that behavior modification is this is not new to us, is trying to modify our behavior and what we should be doing.
In fact, we probably need to rely on the Holy Spirit and the power of God to be able to change our behavior, right?
That's a brilliant idea. So our next question comes from Jackie Mitchell. And Jackie asks, what do you think about the original IHME models for COVID-19? And what is your opinion to date of their huge decrease in the mortality rate?
Yeah, so I think that must be the Howard. I'm blanking on what HMD. I know what it stands for. I just can't remember. I wish Jackie would have put it in her question so that we could actually refer to that model by name and not just by initials. But so like Tony Fauci says all the time on the news, models change because the data that you acquire, you plug that data in and you generate a new model, a revised model. And so I think the models are in fact changing to reflect the type of behavior modification that we've accomplished by social distancing and staying at home. I think that's good. I think the models are as reliable as the assumptions that you put into the models. And so we can only assume that the success that we've had thus far is going to continue and perhaps improve because more people will implement it if they're not already implementing it. And so I think that that's why these models look as though the number of ultimate fatal cases is going to drop. And you have to realize, Jackie and everyone, that the longer we can delay the increase in numbers, the better we'll be, because we have 20, probably 30, maybe even 40 different groups now working fervently to try and produce some type of vaccine. We probably have equal numbers of groups testing different drugs and potentials for inhibiting the viral replication as a treatment. And then we also have people that are trying to sort of upscale the convalescent plasma sera solution as a way to protect people, a passive transfers of antibodies. And so all of those things take time, and they take time not just to develop, but they take time to test to make sure that they themselves are efficacious and safe. And so the longer we can draw this out, in one way, it creates perhaps more frustration because we have to stay at home and we have to be physically distanced a little bit longer. But that's really for our benefit as a whole, not just as a community or as a country, but as a world. And so that's what we're trying to do. We're trying to buy time until we have good solutions. And I hope we can continue to do that.
And just for your clarification, to help you out, AJ, IHME is Institute for Health Metrics and Evaluation.
Thank you, Sia. I appreciate that.
And you can either say it's my brilliance or Google, but
I'm sure Google contributes to all of our brilliance. Yeah.
All right. So the next one, some of these names are just great. So getting through life.
Yes.
I would like to ask this question. Why does the SARS COVID seem to spread more slowly in warmer climates, i.e. the sub-Saharan Africa?
Yeah, I'm not positive that it's spreading more slowly. It may be, you may be right. And that could have something to do with perhaps, and this is a really sort of big leap because I just posted to my Facebook page from the Hopkins Center for Health Security this morning, from their situation update this morning, that there probably isn't going to be this seasonality that we see with flu with COVID. And there may be some decrease in the warmer, more humid climes of summer. But because we see cases expanding and growing, and we have seen that in warmer climates, including sub-Saharan Africa, subequatorial Africa, the southern hemisphere, most of the land masses in the northern hemisphere, but we have seen cases and spreading human to human. The question that you ask, though, is it a slower spread? The answer is it may be. And why might it be? Well, maybe because temperature and humidity are having some effect on the viability of the virus outside the host, its ability to persist in the environment. Perhaps there's an effect there. But one of the other things may be that we are not testing as heavily in some of those countries. And yet, as you might imagine, the health infrastructure in some of the African countries is not fully developed. And so they may not be able to test as many people. They may not be able to report accurately how many cases they actually have. And so we believe that there's probably a lot of underreporting of human to human transmission.
Just kind of wrapping my head around the warmer climate. Now, in my head, I'm thinking like bacteria, which is not a virus. I understand that. But I picture, I admittedly, in my layman self, I almost picture them as equal, where bacteria grows in warm, humid environments. And it seems that viruses, or at least let's focus on this virus. Actually, it's the opposite, right? That it doesn't survive in dry.
So if you think more along the lines of bacterial spores as opposed to living bacteria, then you're getting a better parallel for the hardiness of viruses like this. And so to kill a bacterial spore requires a duration of very high temperature for quite a long time. And so you get one or the other, or a combination of the two somewhere in the middle, either a very long duration at a slightly hot temperature, or a very hot temperature for a very short duration, or something in between those two. And so for SARS coronavirus, the original one, that one was shown to be somewhat susceptible to heat and increased relative humidity. And so they were sort of asking the question, well, because the last case of SARS was in July of 2003. And we think that the reason it went away is because of successful public health intervention and practices like isolation and contact tracing and barrier nursing and barrier protection, and killing the animal reservoir so that it couldn't be reintroduced into the human population. Well, the last case of SARS was in July. And so there was some speculation, did it go away because it got warm in the Northern Hemisphere? And so they did studies on increased temperature and increased relative humidity, which you see in the summer. And what they saw is that you could reduce the amount of infectivity by 1 or 2 logs, which doesn't sound like a lot. And the question is, well, first, maybe you're asking what's a log. So if you started with 10,000 viruses and reduced it by 1 log, you would have 1,000 viruses. If you reduced it by 2 logs, you would have 100 viruses. And so if you have a very high temperature, around 100 degrees or warmer, and if you have a very high relative humidity, 95% or more, then you can actually get a 2 log reduction in virus if you wait 11 to 24 hours under those conditions. And so that's kind of an indicator that there may be some sensitivity to heat and humidity, but what it tells me as a scientist is it's not an end all to be all. It's not getting rid of all of the virus. It has to be a very warm temperature, a very high humidity for a very long period of time. You basically saw no effect if it was as short as 7 hours, or you saw a very minimal effect.
Got it. So what's interesting is I get to see all the behind the scenes stuff here. And so I'm listening to you, I'm seeing the questions, and there is just vibrant conversation happening here. And okay, I'm being made aware that there are a ton of questions.
Okay, so let's move it up. Let's pick it up.
I'll try to pick it up here as well. So great. My apologies. Let's go with Michael Hirsch, and I'm going to read this out loud. And maybe I'm having a hard time understanding, and maybe you can help me out here, AJ.
Okay.
Michael Hirsch asked, would antibody tests of random samples of the population help better evaluate the actual mortality rate and therefore the response? And he also asked, if so, why isn't it being done? Which I don't know if we can speak to that as much as the first part, but
yeah. So it's a great couple of questions, Michael. In fact, it would help. It would help a lot, and especially because we think that easily 50% of the cases could be asymptomatic. So there may be people that have had it, that they've recovered, and if we're randomly testing the population with antibody tests, then in fact we would catch that, and we would grow in confidence that we're getting to that herd immunity, or that sort of saturation in the population of the virus where it's not going to be easily transmitted because it's going to be interrupted by people who are immune. We would get to that level more quickly. And so why aren't we doing that? Well, so the antibody testing has basically been assigned to different states. Different states are handling it by employing universities. The Mayo Clinic hopefully is going to have an antibody test available this coming Monday. And so you want to make sure that they're working, and they're working properly, and they've had enough samples that they get the positives, and they don't call a negative a positive, and they don't call a positive a negative. And to work all of that out again just takes some reiteration, and that takes time. But they've had antibody surveillance testing in Singapore since late February. And so it's easy enough to develop those tests. But it's being done decentralized here in the US. And I know Mayo Clinic's going to have one out soon. I know that there are many other university hospitals and university research labs and companies working on that as well. So hopefully we'll have that soon.
Wonderful. That would be fantastic.
Yes,
let's go on to Muriel. And Muriel Whittam, Muriel asks, if one had a very mild case of COVID-19 with a subsequent immunity also be weaker in quality or quantity of antibodies. So would it be equivalent that a little bit of COVID, a little bit of immunity?
Right. So it's a great question, Muriel. Honestly, it's a great question, and I don't have a simple answer, and that's why some of my answers are pretty long. But the answer is, if a person had an infection, it depends on sort of how robust the infection was. And the only way that you can determine that is to do what's called a titering experiment. So you collect their blood, you take the serum or the plasma out, which has the antibody in it, and then you dilute that serum in serial 10-fold dilutions or in 2-fold dilutions. You basically set up a series of dilutions where each step in the series is a weaker amount of antibody. And so you're actually able to measure how much antibodies in the blood, and you ask, how strong is the titer? And we do that all the time for all kinds of viruses, for all kinds of immunity. Whenever you get a blood test and they want to know if you still have immunity to the mumps or immunity to measles, that's what they're doing. They're doing these titration tests. And what you get back is a number that's like 1 to 48. And that means when you've diluted it 1 to 48, you still have protection. Or you have a number that's 1 to 8. And you have protection at that level. Well, the higher that number, the better the protection, because it means you diluted your serum out a lot, and so you had a lot of antibodies there. And so that lower number is how many fold times you've diluted the serum. And so the only way that we can really answer your question is to do what Michael suggested. We go out there, we randomly test people, and we say, did you ever feel sick? And they're like, no. And you're like, oh, but you have a positive antibody test, and your positive antibody test is at a titer of 1 to 120. And so, that's really good. And so, yeah, so that's the only way to answer your question. And the only way we can do that is to wait, do it, and collect the data and then analyze it.
Yeah, it seems like there's a lot that we're still learning and figuring out on this.
Well, and we're just waiting for it to happen, right? I mean, we can't go out. We couldn't have done this a month ago, right? Because we didn't have enough people in the population at that point that were infected. But now that it's here and now that it's spreading, we can do these kinds of tests, and we should be doing them as quickly as we can.
With you. Okay, Esther Hollis. Esther asks, what might be the reason that we don't know the second animal source for SARS-2 COVID-19? Are we 100% sure that it is the bat mentioned on Eric Metaxas yesterday?
Okay, so what we are Not 100% sure. Scientists are never 100% sure, rarely 100% sure, because we're always collecting more data. And so we always have to have an opening for revising our understanding or revising our models. But, but the data strongly points to this SARS coronavirus 2 originated in bats. That's pretty solid data. I would say my confidence level is 99.5%. And that's based on a lot of different reasons, which I won't go into. But it's not at a similarity level when you compare the genetic sequence of the SARS coronavirus to the bat coronavirus that's most like it. It's about 95, 90, maybe 96% similar. And so you've got this 5% sort of uncertainty. Well, if we look back to the first SARS outbreak, we compared that SARS coronavirus to bat isolates, we saw a lower percentage of identity. And when we compared it to a similar virus that was isolated from something called a civet cat, we saw a very high percentage of similarity, 99%. And some of those changes that were in that 1%, the civet isolate was close to the bat isolate. And so we know that the civet was an intermediate between the bat and the human. We haven't been able to go out and do those tests. I don't know if scientists in China have been doing those tests, but international scientists haven't been able to be on the ground sero testing to see what animals have antibodies to the virus, which would show that they had an infection, or looking for animals that are actively infected and isolating virus from them and sequencing that virus and comparing it to the bat and human sequences. Those are the kinds of experiments that need to be done to answer the question about, is there an intermediate host, or did it come straight from bats into humans? And it's possible. I mean, that degree of similarity, and some of the tests that I've seen where SARS coronavirus 2 can actually bind to the cell receptor in the bat suggests that maybe it came directly from bats. But most people who really study this in fine minutia detail still think that there's probably an intermediate animal, and we just haven't identified it yet. It may be the pangolin, but that's questionable as well. So,
like I said, you're a wealth of information. The fact that you know all of this is amazing.
Isn't it insane how much COVID and SARS information I have in my brain? Yeah.
Well,
I thought it was obsolete information. Who knew it was going to be useful? So
thank you for letting us pick your brain. So Shelby has the next question for you. Shelby Jurgens. She asks, if you have been exposed to SARS-CoV-2 and have developed antibodies, can you still infect other people even though you might not get the disease?
So you could still potentially passively transfer the virus on surfaces, right? And so it's possible, but it's highly improbable that you would, just because the numbers start to really get in favor of the person not getting enough of an exposure to virus. Like you as an intermediate transfer mechanism, you're not going to be very efficient at that, because you're not going to have virus in your body replicating, and you're not going to be shedding virus. The only way that you'd be able to transfer it is if you touched a contaminated surface, perhaps that was still damp and it dried on your hands, and then you shook hands with somebody, or you're like, oh, you've got an eyelash about to go into your eye, and you reached out to clear it, and you touched their eye with your contaminated hand, then maybe you could still infect somebody else, but it would be a very passive mechanism. I hope that makes sense.
Yeah, we're back to wash your hands, don't touch your face.
And don't touch your neighbor's face.
and don't touch your neighbor's face. Kirby Hansen asks a question that...
Kirby!
I know, Kirby, yay! This is so good because it's a lot of people are asking it. Can you distinguish between the symptoms of coronavirus and other viruses such as the flu? And I just want to say that anytime I sneeze or cough right now, and I have asthma, if I go out in public and I cough, I get the hate glares from people. And it's exactly time
Did
to
you see
use
me
the
just wipe my nose? I have allergies too. So
so the idea of gosh, like right now, if you just get a cold,
right.
you're freaking out. Like, what
Yeah, you
am I
are.
gonna die? So
I mean, even I just personal medical information to follow. I was just talking to my doctor yesterday because I'm having an asthma flare right now. And he and I talked through the symptoms very specifically. And I'm like, no, this really is repetitive of previous asthma flares. I'm like, there's no indication whatsoever that this is associated with COVID. And he's like, why are you so sure? And I'm like, well, because I know what all the symptoms are. and so really, because it can be asymptomatic, Kirby, we just it's impossible to say, can we do a systems differential on every case and be successful? The answer to that question is absolutely not. But you can say that if you're going to be symptomatic, one of the most common symptoms, and for the first SARS outbreak, this was 100%. And for this outbreak, it's not 100% because people can be asymptomatic, but for those who are symptomatic, high fever is the clearest indication that you might have COVID. And so what do I mean by high fever? I mean 100 degrees or higher, and sustained. So if you just peak and it goes away, probably not. Maybe it was a mild case, but probably not. If you have a high fever for 2 days, certainly if you have a high fever for 9 or 10 days, you almost certainly have it if your fever is going that long. And the second most common symptom is a dry cough. A dry cough, right? Not a productive one where you're like I am in my asthma flare right now, where when I cough, I sometimes bring stuff up. So again, it's not a dry cough, it's a productive cough. And then some of the other symptoms, which is really fascinating to me, is that people, because this is much more widespread, right? We're talking over 1 million people already infected worldwide, where the first outbreak of SARS in 2003 only infected 8,000 people worldwide. So we have many, many more people with symptoms reporting that they're noticing that they've lost the sensation of smell and taste. And I just think, from what I know of SARS pathophysiology and how it sets up infection and where the virus replicates, that makes perfect sense, right? And so what an interesting symptom. And so I'd say, if you have a fever, if you have a dry cough, if you've lost your sense of smell and taste, there's a really good chance that you have SARS-CoV-2 infection, right? And it doesn't necessarily mean that your symptoms are going to get worse and you're going to end up in the hospital and on a ventilator, but it does indicate that you need to be talking to your doctor and making your doctor aware, and you need to be self-isolating. So
yes, absolutely. That was really, really helpful. Like I said, I think there's a lot of us out here that, any little bug you get right now, it's disconcerting at best. So that's helpful. Thank you. I'm no accountant. They don't put me in charge of numbers, but it's interesting. We've been doing this for 40 minutes now, and we have as many questions as we started with. So I don't know how that works, how the math works, but these people just keep them coming.
Keep them coming.
Keep coming. I think this is from our very own Phil Chin, and he asks the feline situation.
Yes.
If they have similar ACE receptors to humans and can get COVID-19, why is it that human to feline transmission is low or rare, and feline to human transmission is even rarer?
Well, I think that when this is all said and done, Phil, that we're going to realize that human to feline transmission is a lot higher than we thought. And we'll probably never really know the full numbers because we probably won't sero test our cats to see if they converted. But there is indication that if you have a house cat and if you are sick with COVID, that it is possible. I don't know if I want to go as far as to say likely, although I am tempted to say likely, that you can infect your cat. There is one paper out there that has shown definitively that ferrets and cats can be infected with the virus, that they develop symptoms, that they can shed the virus, that they can pass it to other cats, and that they get sick. And I have there's one paper out there And then the situation in the Bronx Zoo with the lions and the tigers, that reported as well. Those cats were infected, were symptomatic. And then there's one guy who's a good friend of RTB who occasionally posts to my Facebook page, Rich. And Rich said that two of his three cats actually were coughing and not feeling well, and they're positive that he's positive that they also had COVID. And so so I think, Phil, that the answer is that yes, human to cat spread is probably going to be more significant and higher numbers than we're seeing right now. Let's imagine we're at the front end of that pandemic as well. The second question you ask is really, can felines or cats transfer it to humans? And because of all of the data I just told you about how they can transfer it to other cats, or how ferrets can transfer it to other ferrets, then yes, probably a cat could transfer it to a human. But let's think about that. If we were sick and gave it to our cat, then there's probably not too many people that aren't going to get it from us, or ourselves included. Our cat's not going to give it back to us, right? So will your exposed cat come into contact with a naive human? Well, it might if you take your sick cat to the vet. So people in the vet waiting room, people who work in the vet's office, the vet, him or herself, might be susceptible and might potentially be infected by your sick cat. But I'm sure that they're wearing masks and washing their hands and not touching their faces. So.
Exactly. It's the trinity of the COVID virus to fight it off. It's wash your hands, wear your mask, and don't touch your face or your neighbor's face.
Or your neighbor's face.
Or your neighbor's face. Again, there's just so much going on right here. I just love the interaction. This is fantastic. Questions are still coming. I'll give you the next question, but I'm also hearing that we will probably run out of time before we run out of questions. I think that that's a given. And they're encouraging us to let people know that if their question, if we don't get to it right now, that we will address it afterwards. And there is RTB Live Extra, which is our podcast version, and it will be out on Saturday. So we will.
So you're basically not going to let me out of the chair until all the questions are answered, but we're going to cut away from live, and some of it will be available in a separate podcast on Saturday called RTB Live Extra, right?
Yes, ma'am. Until you eat all your vegetables, you're not getting up from the table.
Okay, great.
All right. So Harold Chisholm asks, how long can COVID-19 live on surfaces, metal, plastic, cloth? So the different types of surfaces, how long can it survive?
Yeah, so Harold, there's only one study out on that, and it's limited materials. And as I said, it's a single study. And so you'd like to see some independent studies, some repetition of data. But that study suggested that the surface that it dies on most quickly is probably copper. And if it's on copper, it really doesn't stay viable for more than 4 to 8 hours, depending upon if it's this one or the previous SARS. Cardboard was a little bit sketchy. They saw that it persisted there for hours to days. But harder surfaces like steel and plastic, it persisted in this study for at least a couple of days. Previous studies with the previous SARS coronavirus indicate that virus on hard surfaces can persist for weeks. And so 7 to 10 days easily. And so that's why environmental hygiene is so important. And what I mean by that is wiping surfaces down that are touched frequently. And my strong recommendation all comes from what we practiced in the biosafety level 3 laboratory to decontaminate surfaces to inactivate virus. And so the best way to do that is with a 10% bleach solution. And so you can mix that up at home, 1 part bleach to 9 parts water. And that's good for a couple of days because the hypochlorite concentration loses its integrity over time. So you want to make it up fresh every other day, but just spray surfaces down with that. And by the time that dries on the surface, the vast majority of the virus should be inactivated and no longer be able to be transmitted. But bleach can be a little bit corrosive, especially if you're doing this on a daily basis, which is what I encourage everybody to do. Do it on a daily basis, at least once a day, maybe at the end of the day before you go to bed. Wipe down surfaces. Because it's corrosive, after you let it air dry, because it's that time of contact that's actually inactivating the virus. After you let it air dry, come along and wipe the surface off with something else. maybe a Clorox wipe or an alcohol wipe or another kind of disinfectant to kind of not let the corrosiveness of the bleach sit there overnight. Yeah, so that's my suggestion.
Good. Wonderful. We have a question from Chong Ho Yu. And Chong says, it seems that the mutation of the coronavirus is very fast and it's highly adaptive. Is it unprecedented?
So yeah, Chong used a lot of words that I would want to pick apart if we had a long time to talk about his question. But coronaviruses, as a general rule, can undergo recombination and they accrue mutations rather quickly. And just because the coronavirus is accruing mutations doesn't mean that the virus biology is being affected drastically. And so you have to have certain key mutations that take place for it to alter the biology of the virus in a way that's meaningful to us as far as controlling the virus or developing a vaccine or having to worry about whether you can become reinfected with the same pandemic. That's like, if this wave goes all the way around and comes back around in another 6 months, can you be infected because the virus has changed enough? That type of change we don't see that quickly with other coronaviruses. And so the viruses that we're seeing now, although they have, let's say, multiple mutations, you're talking about very large genomes, 30,000 bases of an RNA virus. And so maybe you're catching a dozen different mutations after you've tracked the virus through the human population, maybe 6 or 7 transmissions of a particular strain. And those mutations don't seem to be affecting the ability of the virus to be neutralized by antibodies that were generated by somebody else who had a different mutated virus in them. So yes, coronaviruses accrue mutations, and that's probably why, well, it's one possible reason. Sorry to be so cautious. It's one possible reason why immunity isn't durable. But we also think, based on experiments from MERS, that immunity may not be durable because the virus may be doing something to tamp down memory aspects of the immune system. And so these are great questions that scientists can't wait to answer, and we will be in the months ahead.
Wonderful. Just so much to learn through this whole thing. The next 2 questions are great. I'm hoping that we have enough time to go through a few more of these.
Sure.
Angelica Gonzalez asks the question, why does the COVID seem to affect men more than women?
That's a great question, Angelica. And honestly, if I knew the answer to that, I would probably be making a lot of money somewhere for some. I don't know. We don't know, right? And so again, it's another great question that scientists are eager to explore. I mean, we could make all kinds of speculations. It might be a different concentration of ACE2 distribution in male tissues for physiological reasons that have never been explored before. And to me, I'm sorry to get excited, but I sometimes do when I start to talk about viruses, because we learn so much about our own human biology and about cell biology because of what viruses do. And so, this one right now, it's an international concern, and we grieve deeply for those who are impacted in various ways, not just those who lose loved ones, but those who have to be in isolation away from their kids and their loved ones for prolonged periods of time. My mother's in a nursing home and she can't have daily contact with anybody in the family, and it's been that way for weeks, and it will probably be that way for weeks more. So I don't want to downplay that, but the questions that come up as we try to understand what's going on, as we take those questions into a laboratory and we start to ask, is it because maybe the virus has some type of sensitivity to female hormones that are higher than male hormones? I mean, that's a very practical question we could go look at. I don't have any idea why that might be true, but maybe it is. And if we go in and ask the question and we see that it is, then we ask, well, why is it that way? And in asking those questions and finding the answers, we learn so much about our human biology and physiology and cell biology that it's really, even maybe it's one way that God redeems parts of these types of situations by allowing us to go in and discover things that we can then, from that knowledge, do something to intervene to prevent similar situations in the future, or turn it to good and actually take a virus and make a cancer vaccine out of it, right? Turn the virus into a mechanism that now targets specifically cancer cells, so the immune system attacks the cancer cells. And so we've been able to do those sorts of things. So it's fascinating to me, but it's also just something that, again, it takes time. But scientists want to know those answers as badly as you do, and I just don't think any of us have a clue as to why men are more susceptible at this point.
Your answer was so much more brilliant than my mother's. When I had the conversation with her, she says, because our husbands don't want to wash their hands.
Oh, well, you know what? Your mother may be right. But that's the difference between the perception of a mother and the perception of a scientist, right?
There you go.
And that's why we have to have conversations, so we can bring all our ideas together and look at all of them. So
Exactly. Now, Jessica C is asking a question that so many people are asking, and this really is hitting the heart of many people right now. Jessica C says, thinking of pregnant mothers at this time,
yeah.
are newborns of infected moms born carrying the virus? And are there any major risks for newborns in general?
So the answer to that question, based on what I've seen in the literature so far, is that it does not appear that the vast majority of newborns are at risk, and that there have been newborns delivered through natural birth and through C-section to acutely infected mothers, which means they have infection at the time of delivery, and the newborns are not infected. And so there are sporadic cases where the newborn has been ill, and I think at least a couple where the newborns died, but it hasn't been clear that there weren't underlying issues, sort of what you may hear in the news as comorbidities that were affecting the outcome. And so we just don't have enough numbers to be able to answer the question, was it COVID, or was it COVID plus something else? Or was it just the something else and they also happened to have COVID because that predisposed them to an infection that otherwise they wouldn't have been able to sustain? And so those are important questions. Right now, it doesn't seem like there's any risk to the unborn child. But again, we're only 4 months into this pandemic. And so what we don't know is if women are infected in their first trimester and then they go full term and deliver, whether or not that's also going to hold true, right? And a lot of viral susceptibility to unborn children is susceptibility early in pregnancy. And this just hasn't been going on long enough for us to have that kind of data yet to be able to address that question.
Yeah. My daughter is expecting and gonna have our first granddaughter in June, and I know that this has been on my heart too. And for us as a people, what we can do with these pregnant moms that are on our minds is really intercede in prayer. I mean, there's a lot, like you said,
Pray always, yes.
Pray always, yes. There's so much to learn, to discover, to gain information about this virus, and thank God that scientists are doing what they do. But at the same time, us as those in Christ, we do what we do, and intercede and keep them on your hearts and on your minds.
Yeah, I'll just say this, hopefully for some level of reassurance and to be a bit hopeful. There's no indication from other coronaviruses, and as I mentioned, there's a couple of other ones that have high case fatality, and so I'm not really referring to those because, again, the course of disease doesn't last long enough, and it hasn't been widespread enough for us to have numbers about first trimester pregnancies. But the other coronaviruses, the ones that cause upper respiratory infection, there's 2 of those that circulate in the human population all the time. And then there's 2 other coronaviruses that are associated with the common cold that circulate in the human population all the time. And neither of those 4 viruses, none of those 4 viruses are associated with any type of known birth defects or poor outcomes for pregnant women. So very different viruses, very different biology, but all part of the respiratory coronavirus family. And so So maybe there's reason to be hopeful that we won't see the same types of effects with this that we saw with, Zika or with like that. So
All right.
dead air. Are you reading important questions? What's going on?
Yes.
Yes.
Yes, I am.
Okay. Good.
John Pinto asked a very, very interesting question. He wants to know, is there any truth to the claims that this virus was engineered and is not natural?
Oh, John, is that his name?
Yes.
Yes, John, thank you for this question. So this is what I actually wanted to address at the very beginning before I kicked it off to open questions. Is part of thinking clearly is really sort of addressing conspiracy theory claims such as this virus is a human construct or it's a lab experiment gone awry. And so I just want to kind of come back to what I said at the very beginning. Ask those two questions when you read something like that. What is the source for this accusation or this claim, and what is the data? And then maybe ask yourself a second and or a third and fourth follow-up question to those two. Is anyone who is a scientific researcher anywhere in the world, or is anyone who is a medical professional anywhere in the world, and are there many people in that category that are backing up this claim? And so if the answer to the first two leaves you thinking, oh well, it's somebody who's a watchdog for biological warfare, or it's someone with a political interest, or it's someone with a political agenda, or it's a bunch of people who are very afraid of what's going on around them. If those are the people that are making the claims, then look at the claim with a lot of skepticism, and then go ask that third and fourth question. Can I find global indicators of medical professionals and scientists who are backing up these claims? And is it the majority of them? And I know that for a lot of people, there's a lot of distrust of research science, but I think science is one of the most noble careers that somebody can pursue. And the people that I know, the vast majority of the people that I know in science are out to seek the truth. And they're out to help people. They really want their research to count. And so I guarantee you that if there was any scientific evidence that this virus was a human construct or was a lab experiment gone awry, we would have been able to detect that. And in fact, I've been a part of a small group that has gone in and looked at some of the sequences to ask that very same question. And there's at least one publication out there that's very well done that says to everybody who understands the data that there is no way that this was a human construct. And the question as to whether or not it's a lab experiment sort of gone awry or escaped, again, there's no indication that that's true whatsoever. And there's really good reasons to believe that it's not true. And so I'd say with 98, maybe the same 99.5% certainty that there's no way that this virus was orchestrated or cooked up or produced in a laboratory. Now, why am I not 100%? Well, because maybe there really are some like deep, dark masterminds who are brilliant people who have been able to construct a virus in such a way that it was able to accomplish exactly what they wanted without infecting them, and to fool professional scientists into thinking that nature in fact did this. It's possible somebody's out there that brilliant, that masterful, and that evil. Well, maybe I've watched too many movies, but I guess it's possible.
You know what? It was a great question. It's a question that I've heard a lot of times. I think that sometimes in our own minds, and I'm not saying this about John, I'm just saying this in general, that maybe we're just trying to make this more interesting than what it is, you know? And
yeah, and I honestly think in the case of this particular question, I really think to spread that type of speculation at this point in time is actually fairly irresponsible. And I'm not saying John's doing that at all. I'm glad he asked the question, very much so. I'm happy to address the question. But to now have sort of a really thoughtful response with somebody who's looked at the data on a very like minutia level scale, and who trusts her scientific colleagues that if they observed anything, we would be screaming the loudest that this was a human construct, right? We would be screaming to shut things down. And the people screaming the loudest would be the scientists.
Absolutely.
And
This,
so,
instead of John posting that as a fact on Facebook, God bless him for like coming here and saying, hey, what's the validity and the truth of this?
right.
So, this is the venue. So AJ, are you good for going 10 more minutes? Maybe
Sure.
Okay,
10 more minutes, 2-3 more questions. That'd be great.
All right, let's do this. And then we got some more after that. But again, we'll do that on the RTB Live Extra podcast. And my understanding is in the show notes that there is a way to get to the link, or in the chat that there's directions on how people can sign up for that podcast.
Yeah, and I think at the end, before we cut to that podcast, that I need to remind people about next week's RTB Live, when Hugh Ross is going to be talking. So I'll throw that in at the end again, too.
Perfect.
Okay.
So Shelby is back, and she asks, is it safe to say that this virus is not going away? Many on social media are saying, when this is all over, ... So what are your thoughts? Will it abate, or will it at some point be gone?
Yeah, it's not, I am confident it's not going to disappear the way that SARS did in 2003. I'm confident of that. So the question is, will it slowly peter out? Or will it wane in the summer months, and will we get treatments and vaccines so that we can help control it? I actually think, based on everything that I understand about the epidemiology of this current pandemic, that this is not going away, that we will eventually have to obtain, on a global level, a community level, what's known as herd immunity. There are a couple of ways, and what herd immunity is that just so many people in the herd, right, in the global population, have immunity that if I have the virus, if I'm one of the unlucky people to have it currently, and are there enough people around me that if I come in contact with them, they themselves are immune, so they can't be a vector to pass it on to someone else? It doesn't mean that on rare occasion, one person like me who's infected in a mostly immune herd can't find one other person who hasn't been infected. And so there'll be that sort of slow, sporadic persistence of the virus in the unimmunized or the non-recovered convalescent, right, person. And if it's not durable, then we would expect that it could maintain if our immunity isn't durable, we can expect that it would maintain a low level of infection in a mostly immune global population. And so right now, we think that the numbers that need to reach immunity for that spread to be stopped from significant outcomes is around 80%. But if we look at seasonal flu, right? Seasonal flu comes back every year, and there's variation in that virus, perhaps to the same level or a similar level that there will be variation in this coronavirus. And so it may well persist just like seasonal flu does. But the good news with seasonal flu is we have, at least in the US, we have 50% of our population roughly that gets vaccinated every year. And the good news, and I haven't mentioned this, and I'm so glad I'm getting a chance to now. The good news is that if you have this virus, the next time you get it, we're almost certain, as long as you don't have other diseases or comorbidities at that future point in time, that the immunity that you have, although it may not protect you from reinfection, is probably going to lessen the severity of the disease the second time around. And so, that's definitely true for flu as well. We have an underlying immunity in the population, so everyone's not as susceptible whether they got the flu vaccine that year or not. And the only time that we have real trouble with that, with influenza, is when the virus undergoes a major change, right? If you have something called an antigenic shift instead of an antigenic drift. And that's what happened in 2009 when we had the H1N1 epidemics. It was declared a pandemic as well, although maybe not accurately so. Anyway, so back to, do I expect this to disappear? No, I don't. But I don't think it's going to remain as scary as it is right now, because right now it's a virus running loose in a completely naive population. And so we're in the next few weeks, maybe the next few months, and I can't project too far. I can't predict too far, because these are just my understandings of epidemiology and watching the curves change just like you are. But thinking about it on a global level, watching the curves in local global areas, um, I think that we will get through this worst part in the next few months. And I think that the fall, fortunately, if we start to see it wane in the summer, we have to wait and see what's going to happen in the fall, because we don't know if the summer is actually going to be like a seasonal effect that doesn't make it disappear, but lessens it, and maybe it comes back with a new fury in the fall. And so it may be a few months that we need to do this. But do I think we're going to be in the same position as we are now a year from now? No, I don't. I think because we're going to be better equipped. We're going to have some of the tools that we need to help contain only the people that are sick and that need to be contained, because we'll be able to sero test and test for the virus both. And so we'll be able to contact trace and isolate responsibly. We'll have tools available for treatment. We may have tools available for vaccination within a year and a half, maybe even less than that, possibly, without cutting too many corners. So yeah.
God willing.
Very long answer. Maybe we can take one more question.
Yeah, actually, we do have one more question here. Perfect one to kind of wrap this up. And then you can let us know more about what's happening next week. And then we can get to everybody else's question for the podcast. So Rod Casey, here's a great question for you, AJ.
I know Rod too. Is that from Missouri? Rod from Missouri?
He doesn't say, so I'm guessing. What are your personal practices these days? How isolated are you staying? And what resource do you most often turn to for reliable information if it's not Facebook and Twitter?
Yeah, those are great questions, Rod. So yes, I live alone. I don't have any cats, so not going to be spreading it that way either. So I stay at home and I don't have much contact, but I do have some contact with other people. And I think that that's okay. I think to have sort of a social network of trusted individuals that you know that they've got your back the same way you've got their back. We're not going to get together if we have a fever, if we have symptoms. We are going to practice personal hygiene and environmental hygiene religiously, independently, and when we're together. We practice some physical distancing. You know, I'm not embracing my friends right now. I'm not getting hugs. I'm actually going through hug withdrawal. It's really acute. I was experiencing it this morning. It's like, oh my gosh, when am I going to be able to hug somebody again? But yeah, so I'm doing my part. I have this bandana that I carry with me in the car, so that if I pull up to Starbucks and pick up my mobile order, so I don't actually have to hand anybody my credit card or money or anything, I'll put that on so that I don't breathe on them. And it's always good to see that the people at Starbucks have their masks on now too, so they're not breathing anything out on me. Yeah, and so where do I go for information? I want to actually split that question into one that Rod didn't ask and then address the one that Rod asked. So, you know, I just, as a Christian, I just think that one of the things that we face isn't just what's the virus doing biologically or epidemiologically, but what is the virus doing to us emotionally, and how are we dealing with that psychologically and spiritually? And so as a Christian, you know, I draw a lot of my comfort and my assuredness that my life is secure from my faith and from the Word of God. And I really have found meditating on Psalm 139 to be a great place to go in times of adversity. And I think that this qualifies as a time of adversity. And why I go there is because I don't know any other psalm that does such a great job of first of all telling me that God is intimately acquainted with everything that I do, with every action, whether I'm sitting down or sleeping or standing up, whether I'm staying at home or traveling. Yes, it actually even says that. So, you know, he's intimately acquainted with everything that I'm doing in every situation that I face. And then it goes on to say that there's no place that I can go. And I believe that that's not just any place physically. I believe that there's no place I can go emotionally or mentally or spiritually, that God is not already there, that he's not present with me, and that he's not waiting for me with open arms. All I have to do is turn to him. And then if you keep reading, that same psalm says this, it says, my days were numbered before I ever lived one of them. And so I know that God has ordained my days. And so I don't have to live in fear of anything, including COVID. But I also need to practice responsible public health precautions. So I'm protecting myself and others, right? But trusting my life deeply and first and foremost to the love and the care of God. And so where do I go for other information? And you said not Facebook or Twitter. And I'm really sad that you said not Twitter.
Wait a minute. Ron Casey didn't say that. I said that.
Oh,
So
you
let's
said
not
that.
put that out.
Okay.
I added that.
Okay. So definitely, where do you go for the best sort of public health information? Absolutely, cdc.gov. That's the Center for Disease Control. WHO.int, that's the World Health Organization. I also personally receive daily situation reports from Hopkins Center for Health Security. You can go to John Hopkins website and sign up for those situation reports if you want. There's also a really interesting map where you can see what's going on globally as far as cases. And here in the US, they track all of that in an interactive map there. So that's pretty interesting. But there really are some good places to go on Twitter. And I would point people to three people that I find particularly helpful. And by pointing people to these three people, I am not endorsing any political stance that they may have. And most of them do a really good job of not bringing politics into it, but we're all a little bit biased from time to time, and sometimes that leaks through. But there's someone named Helen Branswell who writes for Stat News. And if you find Helen Branswell on Twitter, she's excellent. I've been following her for well over a couple of years because she covers Ebola outbreaks and all kinds of infectious disease, not just this. Trevor Bedford, who is at Fred Hutchinson Research Center in Seattle. He's actually doing some of the viral epidemiology, the sequence tracing of how the virus is changing as it spreads through different populations. And he's got great insights into and connections with other people who are giving advice and policy advice on how do we come out of this when the curve bends and it starts to come down. What's the best way to get out of this safely? So Trevor Bedford, and then Scott Gottlieb, who was the director for the Food and Drug Administration, the FDA here in the U.S. He does a pretty good job. He had one tweet where he took a lot of heat the other day, and I kind of commented as well. So he's got some watchdogs who follow him too. So read the comments on his tweets too if you aren't quite sure what to make of this tweet, because the people who comment on these people's tweets are also a lot of them are knowledgeable. Some of them are just like other people that just trying to figure out what's going on. But those are good sources, I think. So
yeah, I just
yes. My page and my Twitter page, because I'm sorting through a lot of that information for you, and I'm also reviewing other articles, other sites, from primary scientific literature to just news flashes and updates and stuff like that. So I blog, reasons.org. I blog theorems in theology. I have 2 or 3. Another one that's coming out today. Another one that's hopefully coming out next Thursday. You can also follow me on Facebook, and my Facebook is AJ Roberts RTB, and you can follow me on Twitter, and that's RTB_AJ Roberts. And so, yeah, I'd love to see you out there and post comments. And if you find something interesting, send me the link. I'll check it out and I'll get back to you, and maybe it'll end up in my next interview.
And also, I just want to throw something out there too, AJ, is that if people are enjoying this and this kind of content, that also reasons.org, we have the Understanding the Coronavirus COVID-19 page where we're constantly updating it with your blogs and blogs from other scholars, with the videos like this, with interviews that you've recently had, and it is constantly being updated.
Yeah, thanks for that, Sia. I had mentioned that early on, but I forgot to mention it again. So thanks for bringing that up again. And it's just coronaviruses at RTB.
It's actually, if they just go to our homepage, reasons.org, they should see it there. It's Understanding the Coronavirus.
Super.
And your Twitter feed and your Facebook feed are also embedded on there. So everything is in one very handy place.
Okay, that's great. Well, I think maybe eventually I'll be coming around and doing an RTB Live again as well. But next week, it's actually going to be the man who started Reasons to Believe, astrophysicist and founder and president of Reasons to Believe, Hugh Ross. And he's going to be talking about climate change. He actually has a new book out on the topic of climate change, And it's my understanding from the powers who be here at the organization that if you send a donation of any amount, they'll actually send you a copy of that book. And so maybe if you do that now, you get the book right away, and maybe you'll even be prepared to ask better questions next week on RTB Live with Hugh Ross. So thanks for tuning in, and I look forward to getting some more of those questions in the RTB Live Extra that'll be playing this Saturday. Thanks.
Sounds great. Thank you so much, AJ. This was fun. Thank you.